Welcome, everybody. I hereby open this academic ceremony in which Tingting Zhu will defend the academic thesis Probing and Modulating Motor Inhibition, Multimodal Brain Stimulation and Imaging Approaches to study Cortico-Subcortical network dynamics of inhibitory control. May I invite you to present a summary of your work and the conclusions of your thesis.
Thank you. Dear Prorector, dear members of the committee, dear supervisor team, friends, family, and guests, anyone who's watching the defense of my dissertation, entitled Probing and Modulating Motor Inhibition, Multimodal Brain Stimulation and Imaging Approaches to Study Cortico-Subcortical Network Dynamics of Inhibitory Control. No matter you're here in person or online. In the following 15 minutes, I will give a brief summary of the dissertation. I will begin by briefly motivating why motor inhibition is such a central motor function. Motor inhibition is a fundamental executive function that allows us to cancel, pause, or withhold actions when circumstances suddenly change. In daily life, this happens instantly. Imagine you are driving on the road, and when the traffic light turns right, you must immediately stop. When the light turns green, you proceed. These actions feel effortless, but they rely on extremely fast and precise brain mechanisms. Importantly, when motor inhibition fails, the consequences are not just behavioral, but clinically relevant. Impaired inhibition is a core feature of disorders, such as Parkinson's disease and obsessive-compulsive disorder, where patients struggle either excessive suppression or excessive movement. So motor inhibition offers a unique window into how the brain balances flexibility and control, and why this balance breaks down in disease. To understand how this control is implemented, we first need to have a reliable behavioral measures. In this thesis, motor inhibition was primarily assessed using the stop signal task. Regarding this paradigm, participants watch a bar racing toward a target line, indicated by two arrows. When the bar reaches the target line, they press a button for response. This is a go trial. However, on some trials, the bar stops before reaching the target line. In that case, participants should not respond, cancel their response. This requires suppressing an already prepared action. Now let's try this together. When the bar reaches the target line, we clap once. If the bar stops early, we don't clap. Ready? Let's go. The first one, good. Next one, yes. And next one, good. That's motor inhibition failure, perfectly normal. What you just experienced is the behavioral logic of the stop signal task. That's exactly why this task is powerful. Stopping is not passive. It is an active, effortful control process. It creates a computation in the brain between a go process, which initiates the movement, and the stop process, which tries to cancel it. According to the classic race model, which process finishes first determines your behavior. If the go trial wins, the movement is executed. If the stop process wins, the movement is successfully inhibited. Importantly, even when stopping is successful, we cannot directly observe the stopping process. Instead, we estimate something called stop signal reaction time, or SSRT. It is estimated by taking the reaction time at the percentile of GO-RT distribution, corresponding to the probability of stop fails, and subtracting the mean stop signal delay. SSRT reflects latent speed of the inhibitory process. A shorter SSRT means a faster and more effective inhibition. This paradigm allows us to capture rapid action cancellation. The next question is, how does the brain implement such rapid stopping? It is supported by a distributed cortical basal ganglia loop. During stopping, these cortical regions rapidly engage the subthalamic nucleus via hyperdirect and indirect pathway. They enable suppression of motor output. This anatomical framework is well established. We know which regions are involved, such as pre-supplementary motor area, right inferior gyrus, subthalamic nucleus, and primary motor cortex, but with still unknown a mechanistic understanding of how these regions dynamically interact in real time. To understand this, we must move beyond static pathways and consider inhibition across multiple levels. At the most fundamental level, inhibition depends on neurochemical balance, specifically the interplay between GABAergic inhibition and glutamatergic excitation. This balance determines how excitable your cortical networks are. At the local level, oscillatory activity, particularly in beta band, has been linked to successful stopping. This transient oscillation may temporarily coordinate neural populations during inhibition. Finally, inhibition requires coordinated communication between cortical and subcortical regions within this loop. What remains unclear is how these levels interact to enable this flexible and rapid motor suppression. My dissertation addresses this gap by combining multimodal brain stimulation and neuroimaging to probe and to cautiously manipulate this dynamic. Specifically, we address three main questions. First, at the neurochemical level, we examined whether GABA-glutamate balance shapes motor suppression in real time. Second, we investigated whether beta bursts actively coordinate inhibition and whether deep brain stimulation restores this dynamic in Parkinson's disease. And third, we tested whether phase-specific stimulation can causally tune cortico-subcortical communication and improve inhibitory control. To probe cortical mechanisms, we used transcranial magnetic stimulation, or TMS. TMS delivers a brief magnetic pulse over the scalp, which includes electric current in the underlying cortex and affect cortical excitability. When applied TMS over the motor cortex, the activation travels down to the spinal cord and ultimately processes muscle response in the hand, which are motor evoked potentials, or MEPs. The amplitude of MEP reflects the excitability of the cortical spinal system at that moment. However, the muscle response only tells us about the output of the motor system. To understand the cortical mechanisms underlying inhibition, we need to look directly at cortical reactivity. By combining TMS with EEG, we can record TMS evoked potentials, or TEPs. Cortical responses are time-locked to the stimulation. Different TEP components are associated with distinct neurochemical mechanisms. Early components, such as N15 and P30, likely reflect excitatory glutamatergic activity. The N45 and N100 has been linked to fast GABA-mediated inhibition from pharmacological evidence. Therefore, TMS provides a non-invasive window into the dynamic balance between cortical excitation and inhibition. Most evidence linking TEP components to the neurotransmitter system, which has been established in the motor system. However, motor inhibition does not solely along on the M1. A key open question is whether similar neurochemical signatures can be observed in higher control order region, such as pre-SMA, and how this cortical excitation and inhibition dynamics change during stopping. That's what we have done in Chapter 2. Using concurrent TMS EEG during the stop signal task, we found that active TMS over both left M1 and pre-SMA amplified TEP amplitudes, confirming effective cortical engagement. Motor evoked potentials varied with the task demands. Motor evoked potential, it is higher, or to say larger, in the response trials than the successful stopping trials. Besides, TEP components during successful stopping reflects increased glutamate and modified GABA dynamics relative to goal trials. To conclude, motor inhibition relies on dynamic balance between glutamatergic excitation and GABAergic inhibition. However, local cortical inhibition alone is not sufficient. Effective stopping requires rapid coordination across cortical basal ganglia circuits. This raises a critical question. How are inhibition signals temporally coordinated between the cortical and subcortical regions? In Chapter 3, we shifted focus to oscillatory dynamics in a clinical population with impaired motor inhibition. Parkinson's disease is characterized by excessive beta band synchrony within this loop, which is linked to the motor rigidity and reduced behavioral flexibility. Importantly, beta activity does not occur as a continuous rhythm, but as transient bursts. They are characterized by their duration and rate of occurrence. The EEG data was collected for all participants during stop-signal task, with each patient underwent both deep brain stimulation on and off conditions. The target here was subthalamic nucleus. We found that in the off-stimulation state, patients exhibited prolonged and excessive beta bursts, reflecting pathological rigidity of the network. When deep brain stimulation was on, the motor inhibition was improved in patients. Beta bursts became shorter and more temporally flexible, closing to healthy level. Critically, shortened burst duration was related to improved motor inhibition but impaired strategic slowing, a more cautious response when stopping might be required. This ability can be improved by increasing burst frequency. This finding suggests that beta bursts act as a temporal coordination mechanism linking cortical inhibitory signal to subcortical implementation. DBS, here, it can restore the cortical temporal flexibility, but shifts the behavioral balance. However, this invasive nature of DBS limits its application, so the non-invasive stimulation came into use in a more general exploration and application. Transcranial altering current stimulation, tACS, it applies weak currents to modulate neural activity at specific frequency. In Chapter 4, we applied dose at beta frequency tACS over the right inferior gyrus and left primary motor cortex while recording EEG during the task. By manipulating the aligned and disaligned phases between regions, we targeted interregional synchronization and corresponding behavior. Importantly, the stimulation frequency was individualized based on each participant's beta peak during successful stopping, allowing us to interact with the brain intrinsic oscillatory dynamics. Both stimulation increased local activity. When in-phase stimulation was applied, the frontal motor connectivity increased, which was linked to the improved motor inhibition. In contrast, anti-phase stimulation reduced this cortical connectivity, which facilitated motor execution. Together, these findings demonstrate that phase-specific beta synchronization coordinates this frontal motor communication. While Chapter 4 demonstrated causal modulation at the cortical level, Chapter 5 examined how phase-specific stimulation reshapes large-scale brain networks using concurrent tACS fMRI. In-phase stimulation strengthened frontal motor connectivity, but this occurred alongside reduced connectivity between cortico-subcortical regions and motor inhibition unrelated network. In contrast, anti-phase resulted in higher local frontal parietal recruitment. Together, Chapter 4 and 5 show that beta synchronization selectively reorganized the large-scale network architecture in a phase-specific manner. To conclude, motor inhibition, it emerged from the coordinated dynamics across multiple levels, from neurochemistry to temporal beta bursts to large-scale networks. The key message of this work is that the causal links between neural basis and inhibitory behavior can be established by integrating multimodal stimulation and imaging. Second, neuromodulation works better when stimulation aligns with the brain's intrinsic temporal dynamics, rather than relying solely on the static parameters. And third, these principles provide the foundation for individualized neuromodulation strategies, moving from healthy participants towards patient-specific application in clinical populations. Ultimately, this work advanced a mechanistic framework for understanding and potentially improving inhibitory control in healthy and disease. Before I close, I would like to express my sincere gratitude to everyone who made this work possible. Many thanks go to the Brain Stimulation and Cognition Lab, my supervisory team, and participants, interns, family, and friends. I would also like to acknowledge the China Scholarship Council for their financial support. Finally, thank you all for your attention, and I will give the floor back to the prorector.
Thank you for your very nice presentation, also nicely on time. The opposition will now be started by Professor Poser, who holds a chair in MR methods for neuroscience at the Department of Cognitive Neuroscience, FPN, our university. The floor is yours.
Thank you, Rector. Yeah, dear candidate, thank you, first of all, for the nice summary of your work, which I think was very clear and on time, to say the least. With somebody in a background of MR methods, I'm a little alien to your topic, and I've really enjoyed reading your thesis with a very different perspective, I suppose, and it's given me a lot of new insights that I previously didn't have. So 4 nice chapters, all different focus and different methodology that we will hear about in the different questions, surely. But I first of all would like to congratulate you on this thesis and what you've managed to put together. And of course, the congratulations also go to your team, Inge, as your main supervisor, of course, and Alexander Zack. Now, as said, as an MR person, I felt most strongly drawn to chapter 5, which I thought, oh, wow, they used MRI as well. That's great. Which followed all the other methods. I mean, you basically went through this whole matrix of combinations for TMS, EEG, DBS, EEG, tACS, EEG, fMRI, EEG. And of course, it was nice to see for me again what the additional value of fMRI was, with the additional spatial resolution and information you can extract from that maybe the other methods don't afford you. And as you also summarized in your talk just now, your findings demonstrated that mode inhibition is not just something that occurs in isolated cortical regions, but actually happens much more deeper in the networks as well, which is something that you could quite nicely study with the fMRI. Now, of course, that gives you insights that are important, surely, but the use of fMRI is also something which in practice is maybe not so nice for your subject. It's loud, it's time-consuming, it's expensive for you. And despite all the insights you've gotten from it, I have wondered whether you could use it to infer similar conclusions from simply using tACS in combination with the EEG, even though you mainly study what's happening on the cortical surface, whether the knowledge you've obtained in chapter 5 could be used to perform experiments more like what you described in chapter 4 and hugely simplify the setup of your experiment.
Highly esteemed opponent, thanks for your question. Exactly, the chapter 5 is based on the foundation built in chapter 4, because for the EEG and the fMRI, as you mentioned, they are performed differently with a higher temporal resolution or higher spatial resolution. But I think if I want to simplify the design with MRI, for now, I think we have already simplified it for this physics. Otherwise, we'll do the trypsin with the EEG, MRI, and tACS. But yeah, if I'm thinking about it by using a simplified version, maybe I'll try to to test the different targets, whether the effect were different or the same or not. But for now, I think maybe for now it's already good for the ret IF3 and M1 stimulation, because it really performs the initiation and the end part of this pathway so that it modulates the full network. In the future, if I want to do that something else, I think maybe I can do to check whether the different setup for the parameter of the tACS or the something related maybe work for the effect. But I think normally, based on our finding, the conclusion might not be changed much.
I see.
Yeah.
Okay, but I mean, in a practical setting, of course, ultimately targeting clinical use of your stimulation, of course, it would be much nicer if you could do that in a simplified setup with just the EEG, for example.
Yeah, exactly. We have done that for the chapter 4, because those are with EEG. And I think it works well, but for the EEG, it has a limitation to understand how the deeper regions works. So I think that's why we added MRI. But yeah, at the end, it's lucky for us to make it.
Okay, so may I then turn that around, what you just said? Can we say that the additional knowledge that we've gained in chapter 5 is luxury data to have, and in a practical setting, you may not necessarily even use it, and you can full well get along with what you did in chapter 4?
I partly disagree, because I think it provides a broader value for us to make this story complete. Because if we only focus on the cortical, I think it's better, it's good for the oscillatory network, how it works between the frontal motor regions, or we can add more EEG electrodes to record the prismae or other regions, whether they are related, but it cannot reach deeper. So for me, I still will keep the mind like to use in fMRI.
Okay.
Yes.
Would you like to even combine all three modalities into one?
Exactly.
EEG and tACS and the fMRI together?
Yeah, that's the initial point for the design, but based on the technical challenge, like if we are in the MRI, we should consider the electrode, it should be safe. The safety issue should be prioritized. And if we put the rubber rings here, and then we want to collect EEG, so we need to have a different EEG setting, otherwise the EEG cap, the electrode cannot touch your scalp, so it will decrease the quality of your EEG data. In the future, maybe we can try, but for now, based on the time pressure of the PhD, so that's the reason why we gave it up to separate it into two projects.
I see.
Yes.
Okay. So more generally, from what you've found in those 4 chapters, and I believe they were also conducted in that chronological order, is that right? Or was it reshuffled to make a red line? Either way, I mean, taking the sort of the general finding of it, and the ultimate goal to apply this clinically.
Yeah.
Where do you see the next steps?
I think in the future, as you said, the MRI may be like too expensive for the clinical population. For the EEG, it may work better. But I think it still has a challenge for the tACS application in clinical population, because we need to really ensure we are doing it correctly for the technical part. And also for the patients, I think they work differently from the healthy participants. So that's why we should also reconsider whether the current setup would be the best workout for the patients. So I think before we go in that, we need to still like to have optimized our setting. And also for the clinical use, I think it goes slowly. So for now, I think the EEG one would be good for that.
So how far you think are we from having a little pocket device that people strap on the forehead, sit on the couch with a simple EEG maybe going on, and then apply tACS themselves at home for like half an hour before they go to bed?
I hope it can get as fast as possible, but yeah, reactively, I think maybe it should go at least for like longer than 2 years for broader use. But for now, tACS has to be already applied to the clinical application for stimulation, but maybe with the simplified EEG recording or something else, but not such as the lab, such complex setting. Yeah.
I see. Thanks a lot for your answers, and I give the word back to the prorector.
Thank you. The opposition will be continued by Professor Mess, who holds a chair in clinical neurophysiology at FHML, who is also associated with the academic hospital. And the floor is yours.
Thank you. Would one of the paronyms please collect this envelope? Okay, keep it. Dear candidate, you did not grow up in the Netherlands.
No.
Neither did I. And when I came here, one of the things I learned was there are no problems. There are just challenges. And I really like that. And your thesis was definitely a challenge for me, but definitely a pleasant one. It made fascinating reading, and I learned a lot. So congratulations on this very fine piece of work to you, but of course also to your supervisors. So now let's go to chapter 3 for the questions. First, take a look at table 1 on page 87. Well, normally these tables are pretty boring. This is the demographic, the information on the demographics of the patients. However, in this case, this is not true. This is very interesting. You now may open the envelope I just gave to one of the paronyms. It's a brown envelope.
Yeah.
Open it, take out what is inside, and take a look at the graph. Well, here you can see the degree of bradykinesia in PD patients before and after DBS. Before is orange plus blue, after is just blue. What is your interpretation of this graph?
Let me check.
Yes.
Highly extreme opponent.
It's okay. So you have the 14 patients on the x-axis, on the y-axis, you have the degree of bradykinesia. Orange and blue is before DBS, blue is after DBS. Can you see any differences between the patients?
As we say here, the bradykinesia scores absolutely decrease when the DBS was on.
Yeah, that is the good news, and this is what you want to sell with your thesis. But isn't there a problem? What do you think about the heterogeneity between the 14 patients? Do all have the same degree of bradykinesia?
I'm afraid maybe not or yes, because I remember we did the UPDRS score, and as we found, there might not be such. Did we find, like, as I remember, when plot the bradykinesia scores, they may be close for the patients, okay. but we didn't do the statistic on it.
If you look at these data, for example, look at patient number 3.
Yeah.
The score before DBS is even lower than for patient number 1 and 2 after DBS. So this means you have quite a heterogeneity between your patient group. Do you think that this might have repercussions for your data?
In that case, I think it's not only the patient 3, also 5, 7, 9.
Yes?
Yes, exactly. In that case, if I get a data set, I will do subgroup analysis for that to see whether there's some valuable insights for us.
Yes. I think if you do not have to do that right now, but if you look at table 2 from chapter 3, you will see there is a huge standard deviation in the patient group. And this, of course, reflects this heterogeneity. And I just want to help you. I think that not performing group comparisons, but individual comparisons in the that might be more helpful to see if there is any meaning of these beta bursts. But let's go now to the beta activity, and I really like that. So you have a proposition, and then you are talking about sharpening your tool is important. Okay, so use the tool in the right way. Okay. First question, very simple. What is the frequency range of beta activity?
Normally, we define it from 13 to 30.
That's completely right. Why is that? Where does that come from?
Oh, that's a logic question.
Yeah.
Basically, I would say I have no idea about it, but I think it has been grounded in the previous findings of who defined it.
Yes, you're absolutely right, and that is 100 years ago. So what is the second letter of the Greek alphabet? That's right. Okay, so this is the second rhythm Hans Berger detected 100 years ago, and this is why he called it beta. What is the reason why 30 Hz is the upper limit of the beta range?
I'm sorry, are you asking what?
So the beta range, you said, is 13 to 30.
Yes, exactly.
Why does it stop at 30? Why doesn't it go higher? Is there higher EEG activity?
In that case, I think it reminds me for this project, because we defined the beta as 13 to 30. But exactly, we did another project for this. We're thinking whether the 30 and the 30, they make differences for the beta band. So we defined it to low beta and high beta, except in this range, not out of this range. And we found they did perform differently. For example, in a network view, the lower beta frequency, it may be more related to DBS effects. But for the higher frequency, it might be more related to the behavioral changes. So I think maybe the definition of the frequency is not perfect, but for now, for the anomaly usage, that's how we conclude it.
Yeah, this is already the answer to my next question. This is indeed what I wanted you to think about, that it's quite a large frequency range, and maybe there are sub clusters and that convey different kinds of information. Okay. Okay. Now let's go to page 93. There you have figure 8. Maybe you can have a look at it. it was very complicated for me. And but on the very top, you have time segments, and yeah, the time segments are divided into 100 millisecond segments, right?
Yep.
Okay. How long is a beta wave of, let's say, 20 hertz?
So in that case, for per 100, it will be 2.
So think about it. So if you have a wave that has a frequency of 20 hertz,
Yes.
how many milliseconds does this wave last? You can't find it in your figure. This means 20 waves per second. So how long then is one wave?
0.05.
Well, actually, if I'm not completely wrong, that should be 50 milliseconds. Okay, so
Yes.
okay. So if you have segments of 100 milliseconds long, how then can you detect a burst of beta activity? Because you showed us a very nice image showing several waves. I think it was 4 or 5 waves. They will never ever fit into a 100 millisecond segment. I don't understand that.
I get your point. Like, from your point, yes, for each run, it will be 50 millisecond. Yes, exactly. But in the 100 millisecond, as I remember, there may be 2 or 3 bursts.
Yeah, but a burst consists of several beta waves, and one beta wave already is 50 milliseconds long. So it will get very crowded within this 100 millisecond segment.
But exactly the duration of the burst, it might be shorter than like what we counted here. The duration of the, let me check here. So as you see in figure 7, it shows the duration. It might be like for, I get your point.
Okay, I think
Yeah, it will be like about 100 milliseconds.
I think that's the purpose of this whole discussion, that we have a vivid exchange of ideas.
Yeah.
So, okay, maybe just a last question. So
The question will have to be short.
it will be short.
Answer to.
Why didn't you take into account the amplitude of the beta activity?
Actually, we did.
Oh,
Yes. The reason why we didn't report here would be the amplitude didn't show the differences.
okay.
So we think the duration and the rate would have a better characterize for this disease and the stimulation effects.
Okay, thank you. I'll give the word back to the prorector.
Thank you. The opposition will now be continued by Dr. Klooster, who has expertise in brain science, or I would say brain stimulation and engineering, is affiliated with the electromagnetics for Care and Cure lab at Eindhoven University. The floor is yours.
Yes, thank you, Prorector. First, dear candidates, I would like to give my sincere congratulations for your wonderful thesis, and also I would like to extend these to your supervising team. I do know some things about brain stimulation, but I did not know much about motor inhibition, so I learned a lot while reading your thesis. It was actually a really nice read. I was also very impressed by all the methodologies that you used and combined within these theses. That's definitely not easy, so yeah, very well done. And there was also, I really liked the flow throughout the whole thesis, so it was actually a really good read. Well, having said that, of course, I also have some questions. I have a couple of more detailed methodological questions. And the first one is on your TMS evoked potentials that you use in chapter 2. You mention on page 51, if no peak was found within the window, the amplitude at the center time point was defined as the amplitude for that peak. And I definitely recognize this point. I also struggle with that sometimes. You see these very nice peaks and troughs in the literature, but some subjects do just not show them. But I was actually wondering if you can remember how many cases did not get clear peaks, and if there was any rationale to then, well, just take the center within the time window as the peak, and if that's really meaningful.
Halley's team opponent. As I remember, if it correctly, it might be 5 to 7 not identified within a condition, like among all. For example, for each participant, we have the 6 TP components, maybe among all participants, and for this, maybe 5 to 7 or 5 to 10 has not been identified. So the reason why we take this would be the interpretation of the missing values.
Yeah, that's understandable. And then were these missing values like split over the subjects, or did you, for example, also have some subjects where the missing values were from multiple time windows?
I think it's less happened for the all over missing, but for example, for one participant, it has this shape. Maybe the N35 is like such flow between the P30 and the P60, so this has not be picked.
Okay.
Yep.
So you basically say that the amount of missing values was very limited, so leaving out these subjects would not have affected the interpretation of your results in the end.
Yeah, like for this point, I think I feel it. Yeah, if we because of that we remove the participant is such a lucky, and I think we can keep it. Or if we want to just remove this component for one participant or for 2, the statistic effect size would be different among the different TP components. That's the reason why we choose the interpretation method, because we found it's less happened, and we found it's still valuable and still robust results for our projects.
Okay, fair, thank you. Then I was also highly enthusiastic about chapters 4 and 5, where you applied these in-phase and anti-phase stimulation with frequencies derived from individual beta frequency. Though I was actually a little bit surprised that you derived this individual beta frequency from the electrode FCZ, and then you applied the stimulation to different electrodes, so to the right IFG to F6 and the left M1, so electrode C3. Can you elaborate on this decision? Was there any particular reason? Is this IBF mostly present in the FCZ? And yeah, was there any reason to use different electrodes for the derivation of the IBF and the electrodes where you stimulated?
Thanks for your question. To answer this question, I think, as you mentioned, we extract the IBF, individual beta frequency, from the somewhere near the prism. It's between the red FG and M1. So I feel like it may better characterize the beta frequency, beta peak. And the reason why we choose red FG and red M1 would be, as we mentioned before, to select the initiation and endpoint of this network. We assume that if we stimulate the start and end point, it will modulate the whole pathway. And the reason why we choose red FG, not prism A, which perform a similar initiation role of the motor inhibition, would be if we stimulate here prism A and left M1, they are 2 spatial close to each other. So the electric field will be overlapped to each other. In that case, it's hard to interpret how this stimulation manipulates your network and how to separate the effect from both sides.
Yeah, I need to think about it. Yeah, I think, yeah, I can live with that. So did you actually check if there is variation between these, if there would be variation between IBF at these different frequencies, at these different electrodes?
Yes, we did check. As I remember, the IBF, it might be in the middle. For the mediation would be around 20 for among participants, but some of them may be 17, but some may be 23. So it's within the range, like for 20 plus and -3, somewhere here.
Okay. Okay, and then another thing that you did in both chapter 4 and 5, you performed tACS-induced electric field simulations to confirm that indeed your predefined target was stimulated. In chapter 4, you mostly focused on EEG outcome metrics, so no MRI data was collected, and so you did not have individual MRI data available. And then you run these simulations using standard ERNIE models, which would make total sense, if you ask me. And then in chapter 5, however, you did have individual MRI data, but if I then understood correctly from page 146, you still use a normative model to do your E-field simulations. And I was actually wondering, why did you not use this individual data, so then you could have a bit more in-depth information about these electric field distributions within the subjects. And I was also wondering if that could have made any differences in interpreting your findings.
Thanks for your insight. I am exactly doing that now, because based on the time pressure, I couldn't add this part to the thesis. But for now, I'm using the structural image from individual, and then to mark the central site of the stimulation for each part, and then to do the individualized E-field. And then in the future, I want to investigate whether the E-field strength would be related to the modulation effect and behavior, and also how much differences the variable among the participants. That's exactly already in our list, yes.
Perfect. Can I have one final? Okay, a very short question. It's actually a little bit of a follow-up question of the question that was asked earlier already. I think throughout the thesis you showed that there is quite some importance of applying stimulation during a particular phase. How do you ever think this can be implemented in clinical practice? And I'm not referring to combining it with EEG or fMRI, but more referring to what does that mean for future TMS research? Should we always do that combined with EEG, or should we also do it combined with tACS to entrain the brain before we apply stimulation?
Thanks for your question. I think I have already think about this question before. Like, if I choose to find the index for the participants for the clinical population, which would work better? For TMS EEG, exactly, it's really challengeable because for the setup, it's already hard. And also devices are expensive. And also, the artifact removal is hundreds of work. So I would not suggest to now use the TMS EEG in the clinical, but use a beta BERT, which has been linked to the behavioral and the motor symptom, and also shows its pathological evidence. But in the future, I think TMS EEG and the tACS EEG, we can make the portable version for clinical, which would be beneficial.
Yes.
Yeah.
Thank you. I'll give the word back to the prorector.
Thank you. The opposition will be continued by Dr. Schwab, whose expertise is in brain stimulation and motor control, and who is affiliated with the Institute in Biomedical Signals and Systems at the University of Twente. The floor is yours.
Thank you, Mr. Prorector. Dear candidate, let me start with congratulating you and your supervisors on your amazing work. It has been said already, but I would like to stress again that you have done really 4 experimental studies on your own, including a clinical one. So also the methods that you have used and everything taken together, I think really shows that you have done great work.
Thank you.
Nevertheless, I'm not here to only congratulate you on your great work. I'm also here to ask some critical questions. And I would like to start with a question on Proposition 6. Could one of your paronyms come forward and read Proposition 6?
Neuromodulation works best when stimulation aligns with the brain's intrinsic temporal dynamics rather than relying solely on static parameters.
Thank you very much. The question is if you could explain how much your thesis presents evidence for this proposition. So I think everyone would agree in general with the proposition, and we think that evidence is accumulating for that. But could you say what you specifically did in your studies? Did you compare the individualized stimulation to non-individualized stimulation?
Thanks for your question. So for this, I think it derives mostly from the chapter 3 to 5. For example, for the DBS EEG one, based on the different participants, patients may have different pathological characteristics of the beta bursts. For example, if they have the elongated beta bursts, so in that case, we want to modulate it, we need to shorten the beta burst. But it may improve the reactive inhibition, but decrease proactive inhibition. So in that case, if we're keeping the frequency of the beta burst, would also help to maintain your proactive control. So for this, we think, if we can align the brain's beta burst characteristic, so it may work good. And for the chapter 4 and 5, the reason why we draw this would be we already applied the stimulation at the individual oscillatory frequency. That's why we felt, it really works for the neuromodulation. So we felt, okay, it works, that's great. But we didn't compare whether it's different from non-IBF. But from previous study, I think there in a study before, which has concluded that if the frequency is close to the frequency you are doing the motor cancellation, it may be better modulated for your neural signals.
Yes, you started with the study in chapter 3 on DBS. So there, I understand you had the standard stimulation, standard DBS for every patient, but you're suggesting, maybe you could explain this again, you're suggesting to have an individualized stimulation for every patient that would tune single bursts within a patient. Is that correct?
In that case, I think if we are targeting a single pulse, we need to do the adaptive and closed loop stimulation, which has been already investigated in some studies. By using adaptive stimulation, it did help for the motor symptom and maybe modulated the beta burst already. Yeah, I think it's possible and is promoting.
But in how far would adaptive DBS be able to really steer single bursts in their length and duration, or would it rather change average properties of bursts, like amplitude, duration?
For me, I think I hope it can be fast, but for now, based on current study, what they are doing is to catch the burst properties and then to target. But maybe it's do the cluster targeting, but I'm not quite sure about it. Yeah.
Okay, thank you. Then I would also like to move on with chapter 3, actually, where you looked at the differences between DBS on and off, and how the difference in burst duration or the number of bursts correlated with the difference in behavior, of DBS on versus off. But did you also look at how the burst duration or the number of bursts in total, so within one patient or within a group of patients within one condition, would also correlate with behavior?
I think we have tried, but I cannot remember really clear. We also did communicate how much bursts we have identified for a longer duration, for a longer period. And as I remember, it did show the correlation with the motor symptoms. And also, we also tried to, for example, because we are now doing the correlation for each electrode for E6 and E636. We also did the anterior electrodes together, the amount of the burst rate and the posterior, whether it has the correlation with the motor symptom. But as I remember, we didn't find clear correlation.
But you mean the motor symptoms like UPDRS, or do you mean what you specifically looked at, so proactive inhibition?
Yeah, the behavioral performance.
Okay.
Yes.
So SSRT, for example.
Yeah, SSRT.
There it didn't correlate.
No.
So you do see a correlation across the DBS on and off, but you don't see the correlation within one state, so DBS on or DBS off. But how do you interpret it then? So I understood a bit from your thesis that you may think that beta bursts are in general also coding proactive inhibition, for example. But then we would expect, or I would have naively expected that this would also be something that you see within one condition. So for example, within DBS off.
In that case, I think in the chapter 3, there's a supplementary figure, the figure S1, which has shown the correlation between the burst properties and the clinical symptom severity. It's a pity that we didn't find the significant correlation here, but maybe the motor inhibition, it is too complex. It may have different symptoms, but at the first stage, the DBS may prioritize the whole symptom, but rather the subsize.
Okay, thank you. Then I have a short question on the 4th chapter, where you look at dual-side tACS, and maybe you know that I like this very much. And I saw that your M1 targeting montage is a bit very focused, so probably that you have a focal field. Did you check how large the E field actually in M1 is? Because it can sometimes be a bit lower when you're having the electrodes close to each other.
As I remember, it would be close 0.2 to 0.3 voltage per meter.
For peak.
Oh, no. You mean for M1 only?
Yeah, exactly.
Yeah, from the montage, I think we do the dose-side modulation. So on the M1, it will be 0.1 to 0.3 voltage per meter.
Okay. Thank you very much. Then I give the word back.
Thank you. The opposition will be continued by Dr. Van de Ven, whose expertise is in the domain of cognitive neuroscience and learning and memory, associated with cognitive neuroscience, our faculty, our university. The floor is yours.
Thank you. Dear candidate, also my congratulations on this wonderful thesis and collection of work. I'm really impressed with the multimodal approach of things, that you've used various brain stimulation methods in the clinic as well as with healthy individuals, and the overall mastery of the methodology involved, also in terms of resting state measurements before you actually target beta oscillations, and it's quite a sophisticated piece of work. So I'm very impressed with the quality and the methodological rigor you used. I'm particularly interested in the tACS chapter, since this is also something that I'm working on directly. And I wanted to talk to you particularly about these two chapters for the remainder of our defense. To start off, I'd like to point you to two figures. In figure 4 of chapter 4, there's an indication that in-phase stimulation between the two dual sides leads to a large change in beta power in the right inferior frontal gyrus. You use the same experiment setup for an fMRI chapter to compare, I guess, between EEG and fMRI. But there I find that the largest change in activity is also in the right and medial frontal gyrus, but then for the antiphase. So it's as if the two approaches, when you change methodology from EEG and fMRI, all of a sudden show different or even opposing effects. Could you elaborate a bit on what this would mean and why you would find that in roughly the same area, opposite effects of modulation lead to a change in activity?
Hello, esteemed opponent. Thanks for your question. As you observe, yeah, you really read my thesis. Appreciate it. And for the chapter 4, yes, we did found the right inferior gyrus, which is our target, and on the cortex, that's our target, limited target regions for our interest. So we found the increased activity. But in chapter 5, we didn't find the right inferior gyrus significant activity, but it doesn't mean it didn't active. It just didn't reach the statistic level. But for the left inferior gyrus is also being involved in the motor inhibition, but it's not dominated. So I think this event also adds the insight into how this network works for your motor inhibition to support it.
Okay, so it could be because of lower statistical value, perhaps, that it doesn't quite surpass the threshold. Could it perhaps also be that EEG and fMRI actually look at different things? So for example, a change in beta power that you observe in EEG is not observable in terms of fMRI activity, or is this not how it works?
In that case, it might be possible, I would say, because for the MRI, it works with the slower hemodynamic, so it might not catch such rapid oscillatory, like cortical activity, so it might be possible that it causes the result. Yeah. Okay,
so it may be related to how you would use the various methods to observe, I guess, different dimensions of the activity changes that you would introduce, perhaps. Okay. It's too bad that the manipulations of in-phase and anti-phase dual-side stimulation did not lead to a behavioral effect. These two areas were thought to be involved in the task, yet stimulating with a dual-side approach didn't lead to a change in effect. And that left me wondering what your motivation was to use the dual-side approach. One is, of course, to be able to indicate at two different regions an in-phase or an antiphase signature. But was there perhaps another theory behind why dual-side tACS would actually lead to these type of effects?
Thanks for your considerable question. For the non-behavioral effect, I think it might be normal for the healthy participants because they have already prepared well for the task and it's already contact the ceiling effect. And for the stimulation, I think the reason why we choose the dose set would be it's based on the communication through coherent hypothesis. Because if the two regions, they're oscillation are aligned, they may work better together to support your motor inhibition. So that's why we choose to do the stimulation on two sides, by manipulating the phase alignment to manipulate this network and possibly the behavioral output. But maybe the behavioral output, it just first changed your neural signature, but it didn't reach to the behavioral output.
Yes, that's of course indeed possible. Related to the, I guess the state of oscillations involved in these two areas. So it's particularly to approach a network dynamics between these two areas. It would require that at least one of the areas would carry a beta signal, and in terms of this coherence theory, it would also have to align to the peaks or the troughs of another signal in the other brain area. And we know from tACS that the way that you stimulate in an area, or the effect that the stimulation will have, depends very much on what that particular area is doing at a certain moment. So stimulating over an area that is not involved in any beta frequency might result in a different type of effect than if there's already a beta frequency present in that area. So I was wondering, if you stimulate over one area, you already have to sort of tackle the mode of stimulation together with the oscillations in that area. Now you have two areas. Yes. And those two areas then need to align in time quite precisely, because the window is only 50 milliseconds, in two areas that are quite far away, and that themselves are not direct inputs or outputs to anything. So I was wondering if dual-side tACS, when you have to take all of these interactions into account, stimulating over two different areas that then have to align precisely and then have interactions in ways that are hard to measure in terms of pure behavior, would that not lead to lots of uncontrollable interactions? Would that perhaps explain why there are no behavioral effects, that the things that you're trying to impose are not what you are aiming for?
I'm not agree with this point.
Okay.
Because from previous study, like the stopping comment will move from here to motor cortex. It goes pretty fast, like for about 120 milliseconds, it reached to the M1 already. So based on the temporal precision, I think the dose estimation, which we controlled through the central like NI card by putting the stimulations at the same time, I think it did modulate their oscillation together. It did work.
But you're saying a latency of 120 milliseconds, so that at least 2.5 cycles of beta frequency, right? So from here to there, essentially. So how would you be able to actually track this information crossing if you would stimulate over one area? Do you think that there's a way to map out how stimulation either here or either there, or even both, would affect this information crossing?
I'm sorry, I need first to correct what I said before. The 120 seconds, which means the stop signal, the comment will go to the red FG at around 100 milliseconds, and then at around 120 milliseconds, it goes to the motor. So how the signal moves from red FG and M1 would go faster within a beta circle.
Okay.
So in that case, it goes faster than beta. So for all modulation, I think it did work on this band.
Okay. I would like to take this network approach one step further. There was a recent paper, I think last year in 2025, from the group of Varea and Matthew Krause. I'm not sure if you happened to see this paper, but it was quite interesting. It suggested that if you apply tACS over an area, that it's more likely that you would stimulate the further away projections rather than the local ones. So in terms of tACS-based network stimulation, it's more likely you will stimulate something farther away rather than locally. Do you think that this could also perhaps play a role in the way that you would apply dual tACS?
You can provide a brief answer to that elaborate question.
It might be possible.
All right, fair enough.
Yes.
Thank you.
All right, thank you. Tingting Zhu, the time appointed for defending your thesis has passed. The degree committee will now withdraw and discuss the quality of your presentation and your thesis. So I request that you and your company await us here and await the result of our deliberations. Thank you.
Thank you.
Tingting Zhu, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Zak is authorized to confer upon you this academic distinction in accordance with Dutch university law. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Tingting Zhu, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree or certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Hello, Tingting. Yes, Dr. Zhu, before I hand over to Inge for the laudatio, I would like to take the opportunity to be the first one to congratulate you and tell you that I'm extremely happy and impressed by everything you did here in the last 4 years. I'm very happy how the defense went, happy for you, but I'm also a bit sad because I realize that the time comes to an end with you having you in our lab here in Maastricht, because I don't know if I ever told you that directly, but I really like you. You are
Now I know.
yes. Please remember this. No, I think you are a wonderful person. I mean, extremely impressive. You came to the lab, and I think you could have not made a better impression here for all of us. Not only did you work extremely hard, you overcame many challenges. This is incredible science that you did. The whole Corona discussed the high quality of your work, and I know how difficult this all was, and you just always continued working on this. And whenever I met you in the hallways, you were always smiling. You're smiling now. You're always happy. And also socially, I think you made many friends. If I look around, if I hear people speaking about you, so you came to Maastricht and you made this an incredible success. So you can be very proud of yourself. I hope your parents are watching online. So I would also like to congratulate them for having you as a child. We're very happy with you, and we were going to miss you a lot. I'm looking forward to our trip through China in April. And I really, really wish you all the best for your future. I have no doubt that you will be very successful with your personality and skills. And I really hope you stay in contact for a long time. Congratulations, and I now give the word to Inge for the laudatio. Thank you.
Thank you.
All right, Dr. Zhu, I'm allowed to say that. Dear Tingting, but also all our colleagues, friends, and family, both those that are here and watching now in the middle of the night, right? A PhD is not some kind of a crown that you get at the end of it. It's not a finish line, it's actually a threshold. So it marks the moment when someone no longer does research simply to complete the thesis, but actually because they feel responsible for the question and they want to answer it well. And you now have crossed this threshold. You came to the Netherlands on this CEC scholarship, leaving behind your home, language, family, everything that was familiar, and that decision took a lot of courage to do. Building a life here while conducting these, like we said before, difficult experiments, it took a lot of resilience. And you did it all with a smile and also made us smile. You're always happy and always happy to talk and have a social interaction. And from the beginning, you were the kind of glue for our little CEC student group, and that made sure that everyone felt at home and happy and integrated extremely well into the department. So like we said before, your thesis was ambitious, right? Every study was a multimodal, technically complex, combining brain stimulation, EEG, MRI, and that's not straightforward. It takes someone that can work very precise and organized and has the ability to stay calm when things don't work immediately. And I can assure you that happened very often. At one point, though, this was one of the last studies, chapter 5, for the people that were here today. You completed 60 lab sessions in just 2 months' time, while coordinating a team of 4 interns that you didn't just took along, but you actually trained them to be as proficient as you in the lab, which I think they are still grateful for, if I look at Leah here in the room. And so you were not just collecting data, you were creating structure and transferring your expertise that you built up in the last 4 years and strengthening the little group that you built around you. So I think that you can be very proud of this, at least I am. So it's somewhere along the way we came to a phrase between us that we kind of dubbed "stick to the plan." That was not because you lacked any ideas, quite the opposite, I would say. But because excellent science is not only about doing everything that you could possibly do, it's about doing what matters, about trusting your research question and having the discipline to stick to it and answer that. So over time, I'm very proud to see that you learned not to work harder, but smarter. And I will then, in all this time that we spent together, most fondly look back on our trip to Leuven, which was a very prime example of this, where we faced so many challenges to get the MRI experiment off the ground. We actually just drove to Leuven to pick up equipment that took too long to arrive in our own lab. And it was very improvised, very chaotic, but also very symbolic, I think, for our journey together. And so research, it rarely unfolds perfectly. And sometimes you have to cross borders, literally in this case, but also intellectually to keep moving. And I think you've done a very good job at that. So I would also like to address your family and thank them for supporting someone through a PhD from a distance. It requires patience and trust, and their encouragement definitely make this moment be possible. So what stands here today is not to be reduced to numbers or metrics. What stands here today is growth. You're matured as a scientist, and you became independent. And you arrived here in Maastricht with a lot of talent and courage, and you leave as an independent scientist. Wherever that next chapter takes you, remember, stick to the plan, but you might also revise the plan, expand the plan, and design a complete new plan. So never lose the clarity, discipline, and curiosity that brought you here. And I would like to offer you my deep congratulations. I will miss you so much.
Dear Dr. Zhu, also on behalf of Maastricht University, I would like to congratulate you with the degree you have obtained. Following up on Inge's words, I would also like to congratulate your family, who is indeed somewhere in the middle of the night watching how everything went and caring for you. So they're part of this day, and I would like to acknowledge their presence and also congratulate them. The other thing I would like to say is we as a department, we are going to miss you. You are an undeniable presence in the hallways and in the labs and everywhere you are, and you have this aura. We are going to notice that you're not there. And so I would also like to second Alex's words. We would like very much to stay in contact with you for the foreseeable future and longer. And with those words, I'm going to use the hammer because I realized that I didn't use it to open. So I definitely needed to close it. But after that, stay seated because I have some household stuff to still share with you. I hereby close this ceremony.